So a test cross is testing to see if the parent is a purebred, which in the agricultural or show world is an advantage and it's like more money, or is it a crossbreed, right? Is it heterozygous? So so this is determining the genotype of a dominant phenotype, because there's more than one phenotype. So in this case, let me go so I don't give you an example. So in this case, let's say, well, okay, you guys got in my way here, but I like to see. New share. I think I'll just stay on this screen and we'll just go from there. So in this case, what do we got? We got a cattle, and we know that the cattle has a dominant trait, right? So we don't know if they're homozygous or heterozygous, so we can only give them one dominant allele. We're trying to solve for the second allele. That's a question mark. It might look like the number 2. So we're trying to figure out if they're dominant homozygous or heterozygous. The only way we can do that is to breed them against a homozygous recessive individual. Okay, so I'm going to do a homozygous recessive individual, a purebred. That way, depending on the offspring, oops, that should have been a little hard. Okay, depending on the offspring, if they got all of their offspring showing the dominant trait, I would know the parent was homozygous purebred. But if some of their offspring were recessive traits, I would know, because I'm working backwards, right? I would know that that parent was a heterozygous. So only a a crossbreed. Okay, so you'll see some questions with test crosses, and just know you have to cross them with a homozygous recessive individual. Okay, any questions? So far so good. That's pretty easy. These page numbers that are at the bottom, when I first made this PowerPoint, that's like from a big book I use. It's not your book. So it was really more a reference for me, so I know where to go back and find it. So you can ignore those. Okay, so incomplete dominance. Do you guys remember that from 9th grade? We did some incomplete dominance. Okay, so AP kind of, they don't differentiate very well between incomplete and codominance. They usually refer to them kind of the same, but I'm going to differentiate. And with an incomplete dominant, you write your alleles a little bit differently. So you're going to write a trait like, I'm in this question. Is it okay that I'm not toggling back and forth with the PowerPoint? You guys could have the PowerPoint open. Is that okay? Thank you so much, Micah. So in this case, we're dealing with flower color. So I'm going to use the allele F for flower. And then the color, I'm going to superscript it. So R for red, or W for white. So that's kind of a key for you. You would know what kind of dominance you're looking at simply by the type of alleles they're using. So in this case, incomplete dominant, remember, one is not completely dominant over the other. So red will not mask white, and white will not mask red. They will blend. So incomplete is a blend. So red and white, when you put them together in a paint box, they make pink, right? So if you were heterozygous, like this example here, you would be pink. If you were homozygous for the red allele, so the whole thing is considered the allele. And remember, when you're doing Punnett squares, these alleles are basically representing a gamete, a sperm or an egg. And here's 2 different sperms, right? For example. So homozygous, you'll get red. Homozygous W, you'll get white. Okay. So to do this square, what do we got in this one? Pure line sweet peas with red and pure with white. So that's pretty easy. So this is the first parent. You always want to figure out the parent genotypes, and then from that, determine the gametes. So if this one is homozygous red, I'm going to distribute my red alleles, and the other parent was homozygous white. Distribute those. You get a column and a row for every gamete, and then just bring them down. So typically you write alphabetically. So R comes before W. I mean, it doesn't matter for you guys, but I'm just explaining why I write it that way. So all of these come down. Oops. Okay. All of these letters come across. So in this case, what would my offspring be? Pink, red, white.
Pink.
Yeah, they'd all be pink. And if I wanted to do a heterozygous cross, can you guys guess what you would expect? Let's say both parents are, so first thing, find the parent genotypes. If I cross two of those, so the F2 generation, first thing I find the parent genotypes, that's my cross. What would you expect in the offspring?
One red, one white, and two pink.
Yes. So when I said expected, that's chi-square, right? I mean, it's part of that. So when you're solving, for example, a chi-square analysis, the expected value is coming from a classic cross. Okay, so to see if Corbin is right, a row and a column for every gene gamete. So remember, together this is a genotype. It separates through meiosis, remember, segregation, into individual gametes. So what I'm representing here are those gametes. Whoa, I don't know if that got bigger or smaller in your end. So Corbin said we're going to get a red. So bring these letters down, these letters across. We're going to get a white, and we're going to get two that are pink. Okay, I'm going to work on my spacing. Good. So incomplete, they combine into one color. Any questions on incomplete dominance? So this gets into that chapter 12. Chapter 12 has to do with non-Mendelian genetics. I believe, I don't actually have my book in front of me. Okay. So going on, codominance. So codominance, rather than incomplete, they both will show. So now instead of a red flower and a white flower, you would have a red and white polka dotted flower, a red and white speckled flower, a red and white striped flower. It's going to look like a candy cane. Okay, so both of the genes will be expressed together. So a classic example is the roan coloring on agriculture, or like horses are roan, cows are roan. So you can see in this picture, some of the hairs are white. Since I'm talking about it, some of the hairs are white, yeah, and some of them are red. So they didn't blend together and create a pink cow, but some of the hair showed up one way and some of the hair showed up the other way. Sickle cell is like this. If you have sickle cell traits, if you're heterozygous for sickle cell, not all of your cells are sickled. Some of them are normal and some of them are sickled. Okay, if you have AB blood, your blood has both the A protein and the B protein. So they would both show. They don't blend together and create something new. Okay. So pure breeding herd of, can you get a purebred herd of roan? What do you guys think? Is there any way to be purebred roan?
I think no.
No, right. Because roan itself is a heterozygous trait, right? And purebred means you have two of the same. So this is going to be kind of like your incomplete dominance. In this case, I'm going to use two different genes, two different alleles, okay? One gene, two alleles. So for this one, rather than doing the superscript, I'm going to use two different letters, R for red, W for white. So this cattle would be roan. This cattle would be red, but not truly red. I believe you would say, I don't know, in cows they might be called red. In a horse they'd be bay, or bay is the dark ones. I can't remember, but you get the idea. And this one would be white. Okay? So then if you did your cross, let's just mix it up. Let's have a white cattle with a roan cattle. So in this case, bring these down, bring these across. I'm going to get a roan, a white, and a white. So these are still monohybrid crosses, so they're still easy crosses. It's just you have more than two phenotypes possible in the offspring, right? So if you have incomplete dominance or codominance, you're increasing the different variations. Okay, so that leads us to multiple alleles. So multiple alleles, like typically a gene has two forms, correct? A pea can either be green or it can be yellow. It has two forms. Multiple alleles is when there's more than two forms possible. So the same gene might code for green, might code for yellow, or might code for blue. I made that up. That doesn't exist. But that's the idea. Okay, so one gene, meaning one chromosome, one loci. Do you guys know that word, loci? It'd be locus because it's an individual. I ran out of room. But loci for plural. So the spot that you find a gene on a chromosome is its locus. Okay, so all of these would be found at the exact same position on the same chromosome. So it's a single gene. Okay, but we're talking about blood types. So we know we have A, we have B, and we have O. The classic allele for blood typing is with an I. The I stands for immunoglobulin. Okay, so you have the IA, the IB, and the little I. So in blood type, which you may see on test questions, the O allele is the only one that is recessive. So IA can mask, IB can mask, but A and B are codominant. So they don't mask each other and they don't blend into something new. They are both expressed. Okay? So if you have the genotype IA, you will have type A blood, like me. You could also be a heterozygous individual with the O allele, lowercase I is the O allele, and you would have type A blood. Okay? Anybody type B blood? You might guess if you have IB, you are homozygous for B, or IB little I is heterozygous, right? When you're doing genetic problems, they're pretty much like story problems in math, right? You have to be able to interpret what information is given to you. So they're not going to give you the genotype. They're going to describe the genotype. They're going to say a homozygous man for type B blood or a heterozygous man for type B blood. So you need to know what genotype they're referring to. Okay. AB blood would be heterozygous. I'm sorry, codominant. So they might even just refer to it as codominant. So they're both dominant alleles. So you'd have IA, IB, And the only way you could get, O blood is to be homozygous recessive. My dots aren't showing up very well, so I made a slash. That's a little I, little I. Good? Does this seem like old news? Do you need me to solve this problem for you? Okay, I'm seeing shaking of the heads. So basically a homozygous, or it's just a monohybrid cross. You just have to know how to interpret your genotypes. Polygenic may or may not seem familiar. Of course, poly means many, right? Genetics is referring to gene. So different than multiple alleles, not to be confused, because multiple alleles have one loci, one gene. This is chromosome number 4, one spot, okay? Polygenetic refers to many genes. So maybe you have chromosome number 4, you have chromosome number 12, and you have chromosome number, let's go with 26. And they each have their own loci. So now this is going to act like a more complicated cross, right? This wouldn't, if I only had these two, it would be a dihybrid cross, my 16 boxes. If I had 3, I'd have a trihybrid cross. I can't remember off the top of my head how many boxes that is. I would use my laws of probability and do math instead. Okay, so if you increase the number of genes or you increase the number of alleles, obviously there's more variations possible. So hair color, eye color, height, all these things that you look out at the world and you see so many variations, probably going to be controlled by polygenic traits, right? Our eye color is controlled by 2 different traits or 2 different genes. Skin color, if you are looking at the picture for the skin color, I just accidentally clicked it. You can see there are 3 genes, A, B, and C. So it creates a spectrum of phenotypes rather than 2 distinct phenotypes. Mendel, when he looked at the P's, they had 2 distinct phenotypes, right? Non-Mendelian, lots of phenotypes within a range. So you would refer to this as a spectrum. Okay. Do you want me to do one of those? I'll do a dihybrid one. Bamboo page. At least I'm using the paper that goes with my card. I'm gonna use, whoa, I don't want a color wheel, I just want, I move this whole thing over. Okay, so I'm gonna do eye color. Blue is recessive, brown would be dominant. So I'm gonna use big B, little b. And on one gene, and really it's like how much melanin they set down. So let's say one parent is homozygous in the first position and heterozygous in the second position. And they're going to be crossed with a parent. So this person has brown. So they're going to be pretty dark-eyed. In the next one, we're going to make them blue on the first eye and heterozygous on the second eye. So this one is going to be a little bit lighter, right? A little darker than blue, but a little lighter than the browns. Okay, so remember to find our genotypes. We are going to use perhaps the FOIL method, or you just find every possible combination that you can. So basically when this group sorts independently, right? And they make 4 different gametes. That's the idea of meiosis. Remember, you're going to cut the chromosome number in half. So there's 2 B's. You will distribute 2 B's, and you have, okay, I'm not going to do it that way. I think I'll get confused. Okay, so if you were doing FOIL, you have the first of each pair. Okay, so This pair makes big B, big B. FOIL, just like in your math problems. Outer is referring to the outer limits of the whole set. So outer makes a big B, little b. Inner, so the inner of the whole set is 2 big B's, and last is the last of each pair, so that's big B, little b. Okay? So this parent can make these 4 different gametes, and if we were doing a Punnett square, every gamete gets a column, right? This parent, because I just sorted backwards, first is going to be little b, little b. Oh, that's weird. So the first is little b, outer, little b again, inner, little b, big B, last. The last of each pair, little b, little b. So again, for every gamete, you get a column or a row. So that would be how I'd set up my square.
For the last one, you did outer twice. It should be, first one should it be little b, big B, because it's the beginning of both of them.
Yeah, thank you. So I'll just go like that. Thank you. And that would make sense because I'd want 2 of the same since it was homozygous. So now you're going to bring these down and bring them across, correct? You guys know how to fill in the square. They're just sometimes tricky to set up. Do you want me to fill this in, or do you guys feel okay? Shortcuts, if you have duplicates, like I have these 2, they're going to be the same. These 2, those are going to be the same. So the same relationship, if you ended up with 8 out of 16, it'd be the same as 2 out of 4. So if you understand a shortcut, you can use a shortcut. If you don't understand the shortcut, you probably don't want to use it. But that means I don't have to have a duplicate row. Up here, what do I got that's the same? These 2 are the same. These 2 are the same. So again, I don't have to have the duplicate rows. So now my box just became a little bit simpler. I just have to solve this one, this one, this one. Good. Sex determination, that's easy. You guys probably are okay with sex determination. The next slide. XY, XY. XX, XY. Where's I lost my arrow. Here we go. Okay. Trying to clear something, sorry. I'm guessing you guys are fine and dandy with the X's and Y's, yes? Okay, maybe I'll tell you something about it though. First of all, what did we learn earlier this year? We have the SRY gene, right? Comes up about week 7. And if you have the SR gene expressed, you start to form male parts. And if you don't express the SRY gene, then you will continue with the female form. Something about the XX, dosage compensation. Since males don't need 2 X's, they get along just fine with 1. That means that females truly don't need 2 X's for all of their genes to be expressed. They only need 1. So for females, one of the 2 X's is turned off in every one of your cells. Flip of the coin on which X that is. So like this would explain the calico colors on female cats and not in male cats, because they're turning off one of the X's is not expressed in certain cells, and in other cells the other one is turned off. Okay, so dosage compensation has to do with turning off one of the 2 X's. You'll see that come up sometimes. Good on that? I have no idea what my time is on a Wednesday. We're probably getting close. Okay, we got 20 minutes. Okay. Sex-linked trait, familiar, unfamiliar. So sex-linked traits, we're going to once again use our superscripts. Trying to get my slide out of my way. Okay, so sex-linked traits, typically we're only looking at the traits that are carried on the X chromosome. We're not usually following the Y chromosome because that only affects the male, whereas the X will affect both males and females. So usually you will, if you're doing a sex-linked trait, it's on the X. So it specifically means that, you know, you got this one long X and this little short Y. So it means whatever gene, that's what I want. Whatever gene you're looking at is found only on the X. So we're following that, and we're going to say, we're going to use the X to show that it's found on that, and then we're going to superscript the letter that represents the trait. So maybe this is white eyes. If this was a female, she might have, Debbie was recessive. White, I believe, is recessive. So she might have white eyes, or she might have red eyes. She could be homozygous. She could be heterozygous. A lot of times people, first time around, a lot of times people mess up how to write the male genotype. So you're still going to use an X and a Y. And then you're going to superscript only on the X, right? Because this gene is only linked. This is another way of showing linked genes. It's only found on the X, so we only link it on the X. So if the female was heterozygous for red-eyed, oops. I actually wouldn't use two different letters unless it was codominant, sorry. So whatever color I gave him, he's gonna have red eyes now. And then I'm gonna bring it down. All of the Y's, none of the Y's get letters. All of the X's get letters. So I'm distributing the X from dad, and all of the X's going across. This is mom's X being distributed. This is dad's X, or I'm sorry, mom's other X being distributed. So in this case, you have to pay attention to what they're requesting of you. What % of the offspring have red eyes? 1, 2, 3. What % of the males have red eyes? Okay, so the first one was all of them is like 33%. But if it said what % of the males, you would only look, don't look at this side, you would only look at the males, in which case 50% of the males had red eyes. So the squares are done the same as the monohybrid. You write the alleles differently, only give the X the allele, and pay special attention to what information they're asking of you. Are they asking the entire population, or are they asking just gender specific, male or female? Okay. Some traits they commonly work with, color blindness. That's a sex-linked trait. Okay, well, you got it. And hemophilia. Usually you can't distinguish like red or green, and your blood won't clot. Those are two very common sex-linked traits that they'll use in story problems. Oh, shoot. Okay. So do you want me to do the question in the box. One pair of genes for coat color for cats is sex-linked, the GMB codes for yellow, yada, yada. Do you want me to do that one for you? So what are we mating? We're mating a male that's tortoise. Wait, we're mating a black male. So I have to read the whole story problem to see what is dominant, what is recessive. So I'll usually write down when I'm doing this so I don't lose track. I'll write down what information I have. So capital B is coding for yellow, recessive is black. And if I get both, that's codominant, that's a tortoise. Okay, so I kind of need to keep track of my information when I do these. So they say that you have a black male and a tortoise female. And it's only carried on the X, right? So start with dad and mom. Ooh, you wanted that to be an X. Then that's an X. Okay, so dad is black. I only give my allele. Ooh, Miss Snook. I only give my allele to the X. And mom is tortoise, which means she's heterozygous. So first step in solving genetic problems, always determine the genotype of the parents. Second step, distribute the gametes. Okay, that creates my box. Now, the third step is just carrying them over and down. So these are going to be males. Bring mom's gene across. This male is black. This male is yellow. No, this first one is black, yellow. The second one is black. It's dumb that they use a capital B. And then bring dad's X's down. He's donating a black allele. Mom's donating a dominant and a, recessive. Chalkboards are easier because you can wipe them. So in this case, you have a 25% chance of having a female tortoise, a 25% chance having a female black, 25% chance of having a male black, and a 25% chance of having a male yellow. Or a 50% chance of having an offspring that's black. Okay? I appreciate the head nods. So the dihybrid cross, do you want me to work through the dihybrid cross? Seeing a hand. I'm going to try that again because I have a new set of faces. Do you want me to work through the dihybrid cross? Okay, thanks. Ooh, good. Unknown inheritance problem. So you're trying to solve. This one you're trying to solve what the inheritance problem is. Ooh, it's writing while I talk. So, okay, so based on the data, you want to determine if you think this is like codominant. Is it incomplete dominant? Is it polygenic? That's what they mean when they ask what is the inheritance pattern. So we're looking at two traits. We're looking at shape of the body. Oh no, we're only looking at one shape, aren't we? We're looking at shape of the body. So they're oval or they're round. So in the first cross, you have long and oval, and you get pretty much a 50-50. In the second one, you have long and round, and you pretty much all are oval. And the second one, oval, oval, which is recessive. You don't know if it's recessive. You get 25, 50, 25. So basically a 1:2:1 ratio. Can you guess off the top of your head what type of inheritance pattern that is? Is it complete dominance? Is it incomplete dominance? Is it codominant?
Codominant.
Well, if it's codominant, let's give L and O for our traits. So now way I'm going to find out if I'm right is I'm going to do the Punnett square. So in this case, if I cross two, I didn't set up the square. I thought I was setting up. Sorry. Let me. Okay. Long and oval, long and round, oval, oval. So I read the question incorrectly the first time. I didn't read it as 3 options. So it's definitely either incomplete dominance or it's codominant, right? Because you have 3 types possible. So codominant would say that they're both being expressed, whereas incomplete dominant says they're going to blend. So the long, so in this one, let's, well, you're right. Do I need to solve it? So if you had long and oval and you're expecting this to be, because this could get into your chi-square, right? So you'd have to know how to come up with your expected values. So like the long and the oval, if those are your, the long round only produces 1 offspring. So that's how we know which one is going to be the homozygous individuals, right? Because we know L and R only comes together one way. So this is going to be long, this is going to be round, and this is oval. Now I could then set up the rest of my problems. I could do 2 ovals, LR, LR, and of course, because we've done these so many times, we know we're going to get a 2-to-1 ratio. A 1 to 2 to 1 ratio. Long, oval, round. Good. So when you do chi-squares also, just connecting it to Monday, when you do chi-squares, you also have to be able to come up with what is the inheritance problem, or what is the inheritance pattern if I'm going to make a hypothesis, correct? All right, I think our bell. Pedigrees, how are you guys doing on pedigrees? Do you need a review of pedigrees, or are you good with that one? Okay. The shake of the head means don't need to go over it, correct? Yeah, okay. So then what else do we have? Dominant pedigree. You know how to figure out if it's dominant or recessive, if it skips or doesn't skip. Rules of probability. How are we doing with that? So the sum is when you can say or. So like here I have 2 parents and I want to get this from my offspring, right? What is the chance I'm going to get a big H from parent 1? I have a 1 in 2 chance. So if I'm getting a big H from parent 1, I have to get a little h from parent 2. What's the chance of that? It's a 1 in 2 chance. But is that the only way I can solve this and still meet my desired result? No. I could get it that way, or, and this is that sum, or I could get it this way. Little h from the first parent, big H from the second parent. So again, I have a 1 in 2 chance. So the sum rule, you're going to add the total possibility of each individual event occurring alone. This also gets into the product rule, where I multiply the individual events occurring alone. So what is the chance of getting this in particular, big H, little h? To solve that, I have to multiply the chance of getting the big H from the first parent and the big H from the second parent. So for that, I have a 1 in 4 chance. And the same thing for the second one. So multiply if you need them to happen together. Add if it can happen one way or the other. Now, across like this, pretty easy if you were just going to do a monohybrid cross. But if you get into dihybrid crosses or trihybrid crosses, the math is a little bit easier. So what is the chance? Give me two traits. Flowers. We're going to do flowers. And to make my life easy, I'm just going to use letters and not worry about how I write it. So I have red and I have white, and they're going to be, I'm not going to do it. Oh, where's my eraser? I'm going to do it right. There's red, there's white, there's wrinkled, and round. And I'm going to cross this with the same, but I don't want to do a dihybrid cross because that's 16 squares. That's huge. So I'm going to do a square over here for round or red. Okay. And I'm going to do the W's over here. That's supposed to be a little W, sorry. Try to make them big. W's are hard to work with because it's hard to tell. Okay, so this can be easier. So now I'm going to say, what is the chance of being heterozygous for the first one and homozygous recessive for the second? Now I could do that big O 16 squares, or I can use my math. So I need a big R, little r. I have a 1 in 2 chance here. And I need these to happen together, so I multiply. And a little w only has a 1 in 4 chance here. So rather than doing the big square, I could figure out the probability of each one occurring together. I could get this one or this one, right? So I added 1/4 and 1/4, the sum rule, to be 1/2. And I need this one here, 1/4 chance. So the chance of getting this offspring would be 1/8. Isn't that so much easier? Sum rule, product, and product rule. And that leads us with chromosome mapping, which we looked at the other day, that exact slide. We did a little bit of that. That just leaves us with linkage. And I'm not going to do Hardy-Weinberg right now. Oh, good. So just the linkage groups. My cat. Do you guys have one more in you, or do you feel good with linkage? It's kind of the same as your sex-linked traits. This is our last one. And look, I didn't give you guys any work to do because we just did work all out. So with linkage, this is saying these two traits are found on the same chromosome. Eye color, hair color, for example. Okay? Because they're found on the same chromosome, they should not sort independently of each other, and therefore they act as one. Okay? So if I'm crossing these two parents and hair color, so I'm going to go brown, and I'm going to eye color blue, for example. So 1-on-1. So that's one parent, and the other parent, what do we want the other parent to have? The other parent gets, oh, they're going to be heterozygous for brown, and they're going to be, ooh, I can't use two of the same letters, messed up. I'm going to use R then. Y, I don't know, that wouldn't make sense, but okay. Okay. So, so the big B and the little r on this chromosome are going to be stuck together, right? They don't sort. The big B and the little r on the second one are stuck together. They don't sort. So I'm going to write linked genes with a slash to indicate these operate as one. These operate as one. Okay. My bell says, shut up, Miss. We're just going to go real quick. So the same thing over here. On this parent over here, big B, big R, they're stuck together. And little b, little r, they're stuck together. Send them away, little b, little r. Okay, so I would draw that cross like this, big B, big R. I'm looking at these guys right now, little b, little r. So the slash would indicate they're linked like this. They are found on one chromosome and they cannot separate from each other. Okay, so when I pull them down, I'm pulling these ones down now. These stay together. I don't separate them. I'm not going to go B, B, R, R. That would indicate independent assortment. These are on one chromosome. They move together as a linked unit. They're not going to independently sort. Okay, so stick them together. Same thing, I'm going to bring these guys down. Okay, and now I'm going to bring these guys across. So keep them together. And now I'm going to bring these guys across. Okay. So when I look at this together, okay, I'm looking at that one. So I see big B, so they're going to be brown. And then big R, little r, and brown. So they're having brown hair and brown eyes. I don't see anybody that came up. Oh, this one has a recessive. So big B, little b would be brown hair, but little r would be our blue eyes. You wouldn't use an R, but I did in this case, okay? So you would keep them together, use your slash, but read, you're going to read that as one. So this is brown and this is blue. Does that make sense? I'm a little bit scattered on that box. Okay. So when we see each other tomorrow, we're going to talk about how to know if there was a crossover event from this linked slide, okay? Not this particular slide because this slide was a mess, but